So, hello everyone. Welcome to my next video related to protein binding of drugs. So, we were discussing about the volume of distribution and distribution of drugs. So, we have completed absorption topic in pharmacokinetics and we started with distribution, under distribution where the drugs have been distributed, that we discussed already, and also the volume of distribution also has been discussed. So, under this distribution, the drug have ability to bind with two proteins. Either the drug will bind with proteins present in plasma, which is known as plasma protein binding of drug, which in short it is written as in books PBB, which is plasma protein binding. And drugs also have ability to bind with the proteins that is present in tissues. So the drug can either bind with plasma or with tissues, where it binds with the proteins that is present in either plasma or within the tissues. So the plasma protein binding of drugs plays an important role in affecting the volume of distribution of drugs. So once you are administering the drug, for the pharmacokinetics of the drug, you have to consider this parameter also, that is protein binding of drugs, especially plasma protein binding of drugs. Why we have to consider this plasma protein binding? Plasma, that is blood. So the drug will bind to the proteins present in blood. They will have restricted movement to the different tissues, that is distribution will be restricted, metabolism as well as excretion. Why excretion will be restricted? Because the bound fraction cannot be filtered through glomerulus. So glomerular filtration will be affected, thereby excretion of drugs will also be affected. So generally, because of this plasma protein binding, volume of distribution and half-life of the drug will get affected. So in competitive exams, there will be questions regarding this plasma protein binding of drug. What will be the effect of protein binding in volume of distribution and half-life? I will discuss in the next sections of protein binding of drugs. Let's start with protein binding of drugs. What is protein binding of drugs? The binding of drugs with that of proteins. Say for example, this is drug and this is protein present in the plasma. So the binding of drugs with that of protein is known as plasma protein binding. So it is a reversible process. So plasma protein binding of drugs is a reversible process. It is not irreversible binding or complex formation. It is irreversible. It is reversible. It acts as a temporary storage of drugs. So binding of drugs acts as a temporary storage. They gets bound and make a complex of protein and drug, and then the drug may also dissociate from this protein and it releases the drug slowly. So it acts as a temporary storage. So the binding can be done by formation of chemical bonds. So what are the bonds involved in protein binding of drug are ionic bonds. They may form ionic bonds or weaker bonds like van der Waals bond, or hydrogen bonding will be there, or hydrophobic bonding. So generally with the proteins, the drug may form stronger bonds like ionic bonds or weaker bonds like van der Waals bond or hydrogen bonding, or even hydrophobic bonds will be formed between this drug with protein. The main criteria for a drug to bind with this protein is that they should have complementary shape at the active sites, active binding sites. Now what does it mean, active binding site? Active binding site is the site mainly present in the protein. It is the when it is a protein, so the drug cannot bind in all the sites of the protein. It can only bind at the active site. What does it mean, the active site? It has some functional groups which are responsible for formation of bonds. So to make a bond, you need some specific functional groups. That specific functional groups will be present at the active sites or active binding sites. the drug which are having complementary functional group to that of protein have the affinity to bind at that active site. That is about the protein binding of drugs. So mainly what type of plasma proteins will be involved in binding of drugs? So the plasma proteins like albumin, alpha 1 acid glycoprotein and globulins, also lipoproteins, all are proteins in nature are responsible for binding of drugs. So, what all the things will be affected because of this proteins? First of all, we should know the nature of protein. Proteins are macromolecule in nature. Proteins are macromolecular in nature. Also, they are hydrophilic in nature. What does it mean? Proteins are macromolecules. They are large molecules. Proteins are made of peptides and amino acids which are of blocks. They have blocks of amino acids makes the protein a large molecule. That is the meaning of macromolecule. So proteins are macromolecule in nature and their solubility is in water. That is, proteins are hydrophilic in nature. So any drug which has binding with this protein will eventually become a further large molecule which prevents the entry of drug through the cell membrane. So, through the cell membrane, only small molecules can enter. Since protein itself is a large molecule and drug which has been bound with protein, so the bond fraction of drug cannot enter through the small pores of cell membrane. Only free drug can pass through this membrane. So that is the meaning of macromolecule. And another property I said about protein is they are hydrophilic in nature. So the drugs which are lipophilic also, when it interacts with this protein and forms a bond and complex, they themselves become hydrophilic, water soluble. Even the lipophilic molecules become hydrophilic because of binding with proteins. That is the nature of this protein. So proteins which are having their inherent properties will just share their properties with the molecules that they are adding in or binding along with them. So the bound drugs also will become macromolecule and they also become hydrophilic in nature. So we will discuss about the effects of protein binding. So what happens, what all the properties will be affected by protein binding. So obviously when there are protein binding of drugs, when the drug bind with some proteins, they affect the pharmacokinetics of drug, also pharmacodynamics and some physicochemical property. So what all the pharmacokinetic properties that will be affected, which is absorption, distribution, metabolism and excretion of drugs will be affected by this protein binding. And the pharmacodynamic factor, which property will be affected, that is its therapeutic activity, also the toxicity. So pharmacodynamics involves these two, the action of the drug. Either they provide therapeutic action or they provide toxicity action. And in physicochemical property, it affects the solubility of the drug used. So the drug's property, these are all the property of the drug. Drug's property will be affected by this plasma protein binding of drugs. What all the properties? First four is pharmacokinetic properties and next two is pharmacodynamic properties and this property is physicochemical property of the drug. So first property, absorption. Is there any direct involvement of absorption when the drug undergoes protein binding? No, because it is indirectly involved. Because the drug is exposed to these proteins once that drug enters the plasma, that is blood. This steps takes place after absorption, that is during distribution. What is absorption? Absorption is the movement of drug from site of administration to the systemic circulation. Absorption is, that is, the drug is already in site of administration. When the drug enters the blood, it undergoes distribution where it is exposed to these proteins. So absorption has indirect effect in the protein binding will have indirect effect in absorption. That is the pharmacokinetic property, that is first property, absorption. What this protein binding of drugs does? The protein binding will make the drug into bound form. Some forms will be available in free form, and here this is inside blood. When we see here, that is in site of administration or in GIT, we have only free form of drug. So the drug's free form equilibrium will be disturbed. Free form concentration here, that is in blood, it will be less because of protein binding. It is in bound form. Drug is in bound form. The concentration of free drug will be less in blood than that of the GI region. So the equilibrium will be disturbed. So more amount of free drug have to move towards plasma to maintain the equilibrium. So there will be increase in, so what is the effect of protein binding in absorption? There will be increase in absorption. So let me explain. The absorption will be increased because of protein binding of drugs. Why the absorption is increased? Because the equilibrium of free drug in GIT and blood will get disturbed. So more amount of drug will move from GI to the plasma, which will increase the absorption rate, also the extent of drug towards blood. So there will be increase in absorption due to plasma protein binding of drugs. Now what happens to distribution? Distribution means the drug have to enter into various cells. So they have to permeate through the cells. It is in bound fraction. They cannot permeate through the cells. Once it permeates, it enters into the various tissues. That is, their volume of distribution will be large when it enters through the cells. Since it is bound, they cannot enter to the membrane, but it will confined only in the plasma region. So its volume of distribution will be less, small. So the volume of distribution will be less. It cannot enter into tissue. It can be present only in plasma proteins or in plasma. So it will have less volume of distribution. So once it enters the tissues, they show their action and they will be excreted out of body. So it will have half-life also increased. They are not excreted, they are circulating in the blood. So their half-life also increased. Now what is the effect of protein binding in distribution? Volume of distribution will be reduced and the drug's half-life will be increased because of protein binding of drugs. Now what about metabolism? Again, metabolism is again the binding of drug with enzymes. Enzymes again, they have active sites which accommodate the drug in that active site and gets metabolized and it is easy to excrete. But the bound fraction, which will not have that complementary shape with that of that enzyme active site, cannot bind with enzyme. So will have reduction in the metabolism. Now what happens? Distribution is reduced, that is VD is reduced and half-life is increased in case of distribution. Whereas in metabolism also gets reduced because of protein binding. So will increase the half-life. Once the drug will be metabolized, that is, it is suitable form, inactive form, which is suitable for excretion. But now metabolism is reduced, so the active drug cannot be converted into inactive form or the suitable form for excretion. So the drug will be circulated inside the body as an active form for a prolonged period of time and thus increases the half-life. So the metabolism of the drug also will be decreased because of plasma protein binding. and then excretion. What happens to excretion now? Again, already I said the drug in kidney, that is glomerular filtration, is the first step. So the bound drug cannot be filtered through this glomerulus. So the drug cannot be excreted, and again the excretion also will be reduced, leads to increase in half-life. So when we see about ADME, absorption only will be increased because of protein binding. Apart from that, distribution, metabolism and excretion of drugs will be reduced and will reduce the volume of distribution and increase the half-life of drug. Now what happens to the pharmacodynamic property of the drug? That is its therapeutic activity. Only free form of drug, which is not bound form or otherwise unbound drug, will be therapeutically active. That is, to show a drug its therapeutic activity, the drug must be in its free form or unbound form. It should not be the bound form. If it is bound with protein, it cannot show its action. It cannot be used as for a treatment purpose. So the drug must be in its free form. But when there is protein binding, very less free drug will be available for therapeutic activity. So the drug which are free form only will be responsible for therapeutic activity, will reduce the therapeutic activity because of protein binding. If the drug is having high protein binding, then the drug will have reduction in therapeutic activity or the delayed in onset of action. The drug's onset of action will be delayed because free form will be less, so the action, the effective concentration to reach the blood also will be delayed, because of which there will be delayed or onset of action in case of therapeutic activity. Now think about the toxicity. The drug which is protein bound, the free form will show its activity. Even the toxicity will also be shown by this free form of drug. But the drug is bound, less amount of free drug is present. That's okay. But if you have another drug, that is if you co-administer the second drug which has affinity for the same protein, can displace the drug which has been bound with that of protein, known as competitive inhibition. That is, say for example, your drug is bound with this protein. Now it is not showing any therapeutic activity. The free form is showing its therapeutic activity. If this drug comes out, it may become toxic, maybe. And you are administering another drug which has strong affinity to this protein, can kick out this drug outside, and this can stay here in the protein, which may lead to the toxic concentration of drug and hence toxicity of the drug in body. So if there are any co-administration of drug which shows displacement interaction, may lead to toxicity of the drug. Mainly if the drug is having small volume of distribution or rapid onset of action or highly protein binding and has competitive inhibition or displacement interaction, leads to toxicity. Now what about the physicochemical property of the drug, that is solubility of the drug? The drug will also become hydrophilic. Already I said protein is hydrophilic in nature. So once the drug which binds with this hydrophilic protein also becomes hydrophilic, so the drug will become water soluble when it is in bond form. I'm summarizing again. So when the drug is, so what is the topic of this is significance of protein binding or the effect of protein binding. What happens when the protein binding occurs? So when drug bound with protein, these are all the significant effects that takes place in the body. The protein binding of drugs leads to change in the pharmacokinetic, pharmacodynamic and physicochemical property of the drug. So pharmacokinetics, absorption, there will be increase in absorption due to disturbance in the equilibrium of free drug, whereas distribution, it will be reduced because it cannot enter into the cell membrane, so volume of distribution will be reduced, whereas half-life will be increased. Metabolism, again bound form cannot be metabolized, so again half-life of drug will be increased. And excretion, the bound form cannot be filtered through glomerulus, so again it is circulated inside the body and increases the half-life. And about pharmacodynamic property, therapeutic activity will be shown by this free form or unbound form. So that much of free form, unbound form available will be responsible for showing its therapeutic activity. The toxicity may occur because of displacement interaction by another drug fighting for the same protein binding site. And solubility, the drug may become highly water soluble when it bonds with the protein. In my next video, we'll discuss about the proteins, type of plasma proteins which are responsible for binding of the different types of drugs. There are different types of drugs which are responsible for binding with these different types of proteins, and also the kinetics of proteins also have to be discussed, that we will discuss in the further videos. Thank you.